Yes, that looks good.
Oh, fantastic. I can't believe it. So
Okay. So
thank you.
Juan is going to talk about from equation to artificial life in a test tube without biochemistry.
That is
Very
true.
interesting title.
Yeah. Thank you very much. It's a pleasure to be here and also an honor. I knew Gregoire Nicoli very little, but I met him in Madrid in the 1990s through Manuel Garcia Velarde, who spoke earlier today. And I immediately saw the kind of man he was, an extraordinary scientist and also an extraordinary human being, very passionate about science, which is something we share. And then, so that was in the 1990s. I was a high energy physicist and a gravitational relativity, a general relativity physicist. And then I transitioned into the realm of things that you all are doing and complexity and so on. And what I'm going to be talking about very quickly, because I have very little time, and also because the nature of the problem is very big, so you cannot give many details, but I can give some essential details. I'm going to be talking about exactly what you have there, from equations to artificial life made in a test tube and avoiding biochemistry. Why do we do this? Well, the reason why we want to do this is because we're very interested in life, natural life in our planet. And we're very interested in natural life in our planet because of many reasons, of course, but fundamentally because we would like to understand how life came about in the planet and how life might evolve later. And we want to understand life in the context of physics and chemistry. So there are life as we know it today is a hugely diverse phenomenon. There are about 10 billion species only in planet Earth. There may be life in other realms of the universe. And we know very little about the origin of life in our planet. What we do know is that all life that we see is, as it is said in that at the bottom of this particular transparency, is it provides limitless diversity from a limited, a bounded number of basic components. This is something which is in Darwin's Origin of Species book, in the last chapter, in the famous Tangled bank, Tangled in some editions, Entangled in some other editions, where he described the properties of life in a very particular way, but forgiving one of them, which is information managed with chemistry. He at that time he was not aware of that particular question, the importance of information. What we do know is that all life in our planet is based on the same set of principles. It is based on the same set of principles, and all living systems behave like out of equilibrium physical-chemical systems. So it is based on chemistry, on a form of chemistry which we call biochemistry. And of course, biochemistry did not exist at the beginning of our planet, when the planet was formed about 4.5 billion years ago. At that time, life was probably much more simpler than we think that we see today. And we can do analysis and study how the current three main domains of life, bacteria, archaea, and eukaryota, how they all come from some last universal common ancestor. But this last universal common ancestor is traced to about 3.5, 3.6 billion years ago. So people who are studying the presence of life in other parts of the universe, and the Earth in particular, have a lot of interest in understanding at the root of the tree of life what came before. Was there a simpler form of life? And that particular simpler form of life, was it something that we can actually understand with the tools of simpler science like mathematics and chemistry, physics, etc. So one may ask, what are the simple life mimics we can create? And can we somehow implement those simple life mimics in a laboratory? And the answer is first try, then see, and then conclude. And I would try to do that in the next 10 minutes, which I think is what I have left now. So the first thing one needs to do in order to try and understand if one can formulate a model for life in general is to actually understand or precisely point out what you mean by life. Life is characterized as a physical-chemical system. It's an out-of-equilibrium physical-chemical system, which is capable of using chemistry, handling information. This is an important point because information plays a central role in a form of hierarchical description of life, plays a central role, handling information with which the living system is capable of finding simpler molecules in its environment and metabolize those molecules together with managing energy in such a way that the system is capable of producing parts of the molecules which will be used for the systems programmed through the information that is controlling the system, self-replication. This self-replication is, of course, you can also call it, in the case of living systems, self-reproduction. And when you have that, the system adapts to situations in the environment or changes without adapting to situations in the environment, giving rise to evolution, adaptive evolution, such as the one propounded by Darwin, and of course, that we see today, and also non-adaptive evolution, like the one which occurs with mutations and so on. So can you actually represent this using some mathematical way of doing it because life actually represents, integrates, unifies all these properties in the so-called the cell division cycle, in which any living system, this is a super idealized version of the cell division cycle. It's taken from a beautiful book by Andy Murray and Tim Hunt. It's an Oxford University book from 1993. And here, every living system, in the left y-axis, you have the cell mass. In the in the right y-axis, you have the DNA per nucleus. So you do see that the cell mass goes from 1 to 2. It reaches a point where once it is 2, the system divides, and it repeats that. At the same time, the DNA per nucleus starts with a value 1, and then it begins to grow. It stays stationary, and then it begins to grow, jumps up to 2, and then a little bit before the division, the DNA divides. So this is a universal feature of living systems, which provides unification, the unification of the mass, information, and energy processing in life. It is a long story, but you can actually see that these properties can all be integrated in various forms of the Selkov model. I was very happy to see yesterday that people were, or day before yesterday, that people were calling this model the Selkov model. It appears in the literature with the name of the Gray Scott and so on, but I think it should be called like Goldbetter and Company called it the Selkov model. So this is a model with two species. One of them can be say equated with the cell itself, and the other one is the environment, the food that the cell will take. And you can actually look at this model in reaction diffusion. You will see that there are many patterns. You can then increase the possibilities for the model by subjecting it to the effects of the environment modeled by a stochastic term. And this was done, this is the work on the Selkov model, and the patterns it produces was presented in Science by Pearson. And in 2003, we produced in Physical Regulators a realization of the model with noise, with particular noise. Depending on the kinetic parameters that you have in the model, again, I'm showing the same transparency, depending on the kinetic parameters and the amplitude of the noise, the patterns that the system performs are quite different. This is a particular type of pattern. This is another particular type of pattern. And this is another particular type of pattern where you see that there are like domains that self-replicate. And you can even measure in some of these domains, you can measure the entropy, you can measure the mass of the system, and you can see that it actually, there are conditions of the noise in which the system adapts better. And there are also situations in which the noise actually, or the system actually follows a form. If you look at the simulations, follows a form. Here you have it, of a cell division cycle. In the top, in red, you have the mass. And in blue, you have the entropy of the system, the Shannon entropy of the system minus the Shannon entropy, the negentropy of the system. There you have it. And there are many. You can see that there are cycles that there are many cycles that these systems undergo, and many self-replications, and it's extremely interesting to see the simulations. But the more interesting part is that this model is equivalent to a form of the BC reaction, a model of the BC reaction, the Brusselator, which of course rings home everywhere. And it brings Nicolis again into the picture. And so can you actually use this model or something like it to actually boot up a system which is capable to produce, using perhaps the BC reaction, produce from conditions of very high entropy, produce a system which is organized and is capable of showing the properties of any living system. There are a number of difficulties that you need to overcome. One of them is the so-called concentration problem. Of course, the system cannot be very extended. There is a maximum size that it should have, that it must have, because otherwise you cannot control the information. You need a gradient of free energy. And how can this be overcome? How do you create something which is like a vesicle or some object that insulates the outside from the inside? And how does it work? Well, there is a tool that comes to an experimental tool that comes to the rescue, which is called raft polymerization, in which you can use polymerization to actually generate amphiphilic molecules, which will actually give rise to a vesicle, and that vesicle will self-assemble. You can show that if you take certain relatively simple molecules which contain an amphiphilic, a hydrophilic tail, and then a special molecule here, simple molecule, it has about 12 carbons, and then you have monomers which will be monomerized by the radicals produced by, for example, the BC reaction. Then the system will actually begin to accrete. So each of these linear molecules will begin to accrete. This is amphiphilic, this is hydrophobic, hydrophilic, hydrophobic. It begins to grow, and then the system will self-assemble into structures as the growth of the amphiphile proceeds through the polymerization into structures that actually self-organize and self-organize into objects like that. The experiments are done with very simple instrumentation. The instrumentation is almost kitchen instrumentation, and you produce vesicles out of a homogeneous system. You produce vesicles which, like you see here in this, vesicles that self-assemble from homogeneous conditions. The vesicles grow, as you see here. This is about 30 microns. This is about 8 or 9 microns. They grow, they collapse, and you can see the growth collapse, growth collapse, growth collapse. And during the growth collapse episodes of the vesicle, the population in the system changes, and the population in the system increases. And as it increases, the vesicles themselves also experience a taxis, and there is a process of adaptation that begins in the system. The population increases following a Lotka-Volterra type of population law, and what you end up doing is producing systems which are integrating the properties, the basic properties of life. Before finishing, you can also introduce here a form of a chemical computer to actually produce the vesicles. This was shown in a paper by us in Angewandte Chemie in 2017, and produces vesicles which actually self-assemble, are controlled by chemical computation properties of the BC reaction. And eventually you can actually mix different populations which actually compete among themselves. I just wanted to give a few shots of what is going on. And let me conclude very quickly. We set out to mimic simple natural life using an approach which is a top-down or outside-in approach, as they call it in computer science, where starting with the properties of the living system, we actually produced a mathematical model of the same, which corresponds to the equations of the Busselator, as a matter of fact. We are able to implement the basic properties of life, information, for example, or metabolism and so on, with chemistry without using any biochemistry, with small molecules. And we are able to start with small molecules and boot up from a homogeneous mixture, interesting autonomous systems, which make up populations and are able to actually evolve in a very primitive way, and which are capable, as I said, of integrating all these properties. And right now, we are producing a system in my lab where we are able to actually generate molecules, short, small molecules again, which adapt and are capable at the same time of producing, of handling information in the form of some simple, super simple precursor of a gene. Thank you very much for your attention. And I hope that if you're interested, you could take a look at our website. You can find our website is just Google my name and then top down or Harvard University, and you will find all kinds of information on this work. Thank you very much for your attention, and forgive me for being a little later than I wanted to be. Thank you.
Thank you, Juan, for this nice talk. So, any question, please? I see there is, no, I see a clap.
Thank you.
Any question?
Yes.
Go ahead, please. I can.
I have a question.
Yes.
Do you hear me?
Mm-hmm.
Yes. Thank you, Massimiliano. I can hear you.
Yeah, sure.
I'm not Massimiliano, but
You referred several times to the notion of information. So is it the same notion as information by Claude Shannon, you know, where you can measure information, or is it something which is less well defined?
No, this is information precisely in the Shannon
Shannon?
way, in the Shannon way. Information is in the Shannon way, or if you want, Biluan way.
Mm-hmm. It's a kind of entropy somehow, you know.
It's a type of entropy, is the Shannon entropy, which is equivalent to Gibbs entropy. Of course, as you know very well, I don't need to tell this to you. And you can. This is the type of information that you handle. In fact, you can see if you look if you look at the information properties of all living systems, you can see that for bacteria, Shannon information is all contained in what it is called the central dogma of biology, the molecules participating in the central dogma of biology. For you, me, plants, and eukaryotes, only a part of that Shannon information, the channel capacity, channel capacity is implemented. You and me have an extra, a way to actually go to maximum entropy, which is using the so-called non-coding DNA, or formerly junk DNA, which I think is a really bad name. So it is Shannon information that biology uses, and it is Shannon information that the system can handle.
So this is what quite close to what Shannon had in mind with the entropy and the channel capacity, etc. Oh, that's interesting.
Yep. Thank you.
So thank you. Massimiliano, just very short question.
well, my question was similar in these self-organization experiments that you showed us. At what level can you quantify things in terms of this Shannon entropy?
There are two ways of your question cuts two ways. First, at the system level, and that you can do just by looking at the experiments and looking for an appropriate measure for the probability distribution function. And then there is another way associated with it, which is where is the information fed into the system so that the system does this or does that. And that can be quantified by the way in which you feed the system. You see, in these systems, the role played by the DNA bases, when you take the DNA bases and you process them through the ribosome and produce, for example, proteins, the same role as produced by the sequence of DNA bases, which is translated into into proteins, is produced in these systems by feeding aliquots to the system and promoting processing them through the processing them through the BC reaction or a chemical computer that you can put. If you are interested in these things, here is a transparency where you can check that paper. Just it's in iScience, and there you can get some information or write to me, and I'll be more than happy to continue the conversation. Thank you for the question, and thank you for the time.
Thank you very much.
Okay, thank you. So Yanis.
Yes.
You are ready?
Yes.
Could we have, please, Juan, can you please unshare, yes, unshare your screen? Thank you.
Yes, thank you. Thank you.